All right, we are going to start on lecture 14, where we are going to discuss the anatomy and the structures of the brain. So, a brief introduction on the brain. It is a complex three-dimensional structure that performs an amazing array of functions. We often think of our brain as the supercomputer of our body. It consists of roughly 20 billion neurons, and that is actually argued sometimes, and people think that there could be upward of 50 billion neurons within the brain. So let's start by talking about how the brain is organized. I'll give you a brief overview of some of the structures that we're going to talk about throughout this lecture. We're going to talk about, and this is kind of in an inferior to superior order. So we'll talk about the medulla oblongata. That is the region of the brain that turns into the spinal cord. Also on this region, which makes up the brainstem, so the medulla oblongata, the pons, and then something called the mesencephalon, also sometimes referred to as the midbrain. Those regions make up the brainstem. And then you have the diencephalon, and these structures are surrounded, enclosed almost, by the cerebrum. So the brain, the outer portion of the brain, surrounds those regions. So those are kind of internal structures that you'll see. And then the cerebellum. So let me show you what some of those structures look like. We're not going to go into great depth right now. We'll talk about them more as we get to them in the lecture. So you can see here is where the spinal cord would start. This is considered the medulla oblongata. The pons is here. The midbrain, that's here in green. So I'll just write these here for you. Medulla oblongata. I think I have another slide that shows all of this too. This is the midbrain or the mesencephalon. This here in red is the diencephalon. Again, these are deeper structures. So the cerebrum, which is this outer region, is kind of encasing those structures. And you can see the cerebellum here. So we'll systematically work through that, take a tour of the brain, and look at these different structures. Here's a close-up of that internal structure. You can see some of the features are highlighted here and what their functions are. I'm not going to mention those right now. We'll talk about those more in depth as we get there. But you can see the medulla oblongata. Here's the pons. The midbrain is in green, and the diencephalon is shown in pink. And this is what people normally refer to as the brain stem. But we will use more specific terms since we are learning about the brain. So let us first talk about the structures that are going to protect and support the brain. So the brain has a consistency of weak jello, very soft and delicate. It's not going to be like the brain that you're going to see when we do the dissection, because that brain has been fixed, and so it's going to appear to have more structure to it, but that's not actually what the brain is like in a living brain. So because it's so delicate, there has to be several levels of protection. So here are a few of those levels of protection. So first of all, we have the plate bones or the flat bones of the skull that we learned about, right? The parietal bone, the frontal bone, the occipital, and the temporal bones. And then below those bones, beneath that, we have a series of membranes called meninges. And we're going to take a deeper look at those meninges because they are structured a little bit differently in the brain compared to the spinal cord. We did touch a little bit about the blood-brain barrier, And this is just to remind you that this barrier prevents most of the chemicals that you're going to find in your bloodstream from gaining access to the interstitial fluid of the brain. And that's important. And then finally, we have cerebrospinal fluid, which we tend to associate, right, with the subarachnoid space. And it acts as a shock absorber. But we don't form it in the spinal cord, we form it in the brain. So we're going to revisit that here in this lecture. Here is a sagittal view of the brain. You can't see all that much in this image, but you can see the bones, right? And then below that, here are these meninges. You can kind of make out the meninges. And then that space, that dark-filled space that you're seeing, I'm going to take a yellow so you can see it. This dark-filled space right there, that would be filled with cerebrospinal fluid. All right, so let's start to take a closer look at these layers. So this is a great image from your textbook, and this view peels away each of the layers sequentially, deeper and deeper, so that you can see what is covering the brain. So you can start over here with the scalp. So the scalp is going to contain the skin and the hair. And then underneath that is a muscle layer. So if you remember learning about the frontalis and the occipitalis, and that there was aponeurosis that connected the two, so that's what's under there. Below that, you will find some loose connective tissue. And then you will get to the cranium itself, the bone layer, the periosteum. Below that, you will find the dura mater, right? That's that really tough layer. And then this reddish color is the arachnoid mater. And then finally, adhering to the surface of the brain is the pia mater. So here is a close-up where you can see those layers that I was just mentioning. The dura mater in certain locations is going to split into two layers, and I'm going to come back to that. But I would just want to remind you that below the dura mater, you can see the arachnoid mater. And remember, the arachnoid mater has those struts coming off of it, right? You see these struts here. And that is what is connecting to the underlying pia mater here. So that's this layer here. And then running through that subarachnoid space is the cerebrospinal fluid. And remember, we called those struts the trabeculae. All right, let's take a closer look at where and why we get that split of the dura mater. All right, so the dura mater in certain places is going to have two layers to it. It's going to have a periosteal layer, which is a layer that is connecting to the periosteum, which is how it got its name. So that's the periosteum of the cranial bones. And below that, there's going to be a meningeal layer. This is a deeper supporting layer. And in the middle of those, there is going to be blood that is flowing through there. It's actually going to be venous blood that is going to make its way down to the jugular vein. This is how the brain is going to drain its blood. So after the blood, like it's depleted from oxygen and the nutrients are removed, it needs to drain and head toward the heart. And so these two layers are separated by a gap containing fluid and blood vessels. And you're going to see in some instances that these gaps enlarge to form what's called dural sinuses. All right, so this is a closer look. You can see here the dura mater, and you can see it split into these two layers. It splits into the periosteal layer and the meningeal layer. And then flowing through there is venous blood, which will eventually drain into the jugular vein and head back to the heart. In certain areas, the sinuses actually get quite large. So you can see one of these sinuses here called the superior sagittal sinus. This is an enlarged dural sinus. And you can actually see some of the blood vessels that we talked about that are in the subarachnoid space are connecting with these sinuses to drain that blood. I want to talk about another feature of the dura mater. So there are, in certain locations again, the dura mater is going to form folds. And these are extensions in the meningeal layer. So these are extensions of the inner layer, specifically the meningeal layer, into the cranial cavity. And the purpose of these extensions is to stabilize and support the brain even more. And there are 3 large dural folds that you need to know about. So I'm going to tell you about the folds, and then I want to show you them and show you what they're doing. So the first one is called the falx cerebri. The second one is the tentorium cerebelli. And the third one is the falx cerebelli. Let me show you what these are, because I think it's easier to see on an image. Let's talk about the falx cerebri first. So this is a sheet of dense, irregular connective tissue that separates the left and the right hemispheres of the cerebrum. So you can see this in the image right here. This material right here is dividing the left and the right hemisphere. And it's anchored here at the back. So right about here, it's anchored by the occipital bone. And then in the front, there's another little anchor. You might be able to guess what it is. We talked a little bit about it when we were talking about the bones of the skull. It is the crista galli of the ethmoid bone. So that is how this falx cerebri is anchored in the brain and helps to stabilize the brain. The next fold is the falx cerebelli. This one is found in a rather shallow fold between the left and the right hemispheres of the cerebellum, okay? And then finally, the tentorium cerebelli separates the cerebrum from the cerebellum. So that is what's listed here. And then you can see these folds better in this image. Together, these membranes help stabilize that fairly loose material that comprises the brain. All right, let's explore the arachnoid mater a little bit more. So this is the next deeper layer. It connects only loosely to the dura mater. And you remember it has trabeculae, which branch and connect to the pia mater. And that the subarachnoid space, it is filled with cerebrospinal fluid and blood vessels. Take a look at this again so you can see here is the arachnoid mater right here, right? And you can see those struts, those trabeculae here, connecting closely to the pia mater. You can also see blood vessels there, and remember, flowing through here is the cerebrospinal fluid. There is one more feature that I want to show you on this particular picture, and that is something called the subdural space. The subdural space is actually, it's considered a potential space or a virtual space. It's generally not there. So normally the subarachnoid space is filled with cerebrospinal fluid, right? We've talked about that several times. And what it does is it pushes on the arachnoid membrane and kind of shoves it against the dura mater and holds it in place. But if for any reason you lose cerebrospinal fluid, it will pull away. All right, so the other situation is you could have a head wound that penetrates through the dura mater. So here is the dura mater right here. So if you penetrate through the dura mater, that is going to allow blood to pour into this virtual space. And then it'll separate. And what will happen is those two layers will separate. You'll get blood that flows in that subdural space, and you'll get what is called a subdural hematoma. And that could be somewhat dangerous because it starts to compress the brain, right? It's going to push down onto the brain because there's bone at the top and the bone is not going to stretch. So when you push down on the brain, that actually becomes fairly dangerous. And sometimes clinicians will have to drill a hole to relieve some of that pressure. So those are the features that I want you to realize and understand on these layers. All right, this is a close-up showing you. I probably should have showed you the potential space here, the subdural space here, but you can get a better idea. This is a closer look where you see the dura mater, the arachnoid mater, and the pia mater. Here are those nice trabeculae holding it down. And again, if anything should happen with this, if you lose any cerebrospinal fluid, this potential space can get bigger. Or again, if there's injury here and blood flows in there and begins to fill up, it'll start to push this membrane down onto the brain and cause some serious trouble. And then finally is the pia mater. This is very delicate. You can't easily peel the pia mater off of the brain. The blood vessels that enter the brain will also be covered in pia mater. So it's going to continue to follow every nook and cranny of the brain, all the crevices. It'll surround blood vessels. None of the blood vessels are going to be in contact with nervous tissue. So you can see here is a nice image of the pia mater. You can see how it follows along even when there's a blood vessel present, even when it branches, it's going to follow around the nervous tissue continuously. The next source of protection is the blood-brain barrier. And this consists of a single blood vessel, which is made up of simple squamous cells. So what you have are these simple squamous cells. The nucleus is here. And they come together. There's going to be a tight junction between the two cells. And there's tight junctions there because this is a source where you can get a lot of stuff either going in or coming out. And we rely on that in most places of the body to exchange nutrients and waste. And there are a lot of ions in blood that could harm the nervous tissue. So there can't be a free exchange of material. So you can think of this as a long vessel here. So the blood-brain barrier has a lot of tight junctions so that they don't leak. So if something is going to come out, it has to be actively transported by the cell. So let me show you what a picture of that looks like. So again, you can see here these simple squamous epithelial cells, and in between each of these cells here, there are lots and lots of tight junctions outside of the cell. And what this does is it prevents things that are in blood from leaking out. In addition to those tight junctions, if you remember, we talked about this already when we were talking about the spinal cord. So we're just going to revisit this kind of quickly here. But there are those foot processes from the astrocytes that are surrounding those blood vessels. So in this image, it shows that there are some gaps between those foot processes, but in reality, those gaps aren't there. They are also linked by tight junctions. So you would have tight junctions running in between all of these foot processes, kind of stitching them together. So not only does something have to cross the epithelium, but it also has to be transferred by the astrocyte. So ultimately, it's the astrocyte that decides what gets in and what comes out of this blood vessel. So if you remember, we talked also about cerebrospinal fluid, and that it is going to, so we talked about it in respect to the spinal cord. Now we're going to talk about it with respect to the brain. And it is going to completely surround and bathe the exposed surfaces of the brain. So we talked about it being in the subarachnoid space and the central canal of the spinal cord. And this is all continuous with the subarachnoid space of the brain, okay? It is one narrow space. And there is a total of about 150 milliliters of cerebral spinal fluid flowing through those spaces at any given time. And the entire volume is replaced about every 8 hours or so. So this is a relatively small amount of fluid because you move about 150 milliliters of blood with every 2 heartbeats of your heart. But there's no pump for cerebral spinal fluid, so this is going to be much slower. And cerebral spinal fluid is a derivative of plasma. So there are several reservoirs that hold this cerebral spinal fluid and produce it. And these are called ventricles. So the brain has 4 of these ventricles. And they also have a tissue inside the ventricles which make up the choroid plexus. And that together is what is going to produce the cerebral spinal fluid. And we're going to look at that in a minute. So these choroid plexuses are located within the ventricles of the brain. And if you remember, we spoke about ependymal cells in their very first lecture on the nervous system. So here is an image of the brain. So we can see the cerebrum, we can see the brain stem, we can see the cerebellum, but what's in the middle of this is what's interesting. This is highlighting the chambers that are called ventricles that contain cerebral spinal fluid within the brain. There are 2 paired left and right lateral ventricles shown here. So this is left and this is right. Those are the left and right ventricles. There is one on each of the 2 hemispheres. And they are connected by a narrow passageway called the intraventricular foramen. So they're connected right here by this intraventricular foramen. And this leads to the 3rd ventricle, which is right here. So the 3rd ventricle is wedged in between the 2 halves of the diencephalon. And this is connected by a narrow canal called the cerebral aqueduct. That's right here, the cerebral aqueduct. This is the aqueduct of the midbrain. It passes through the midbrain and then connects with the fourth ventricle, which is right here. And there are a variety of passageways that lead down to the central canal. So we're going to go down into the central canal of the spinal cord. And this is also going to lead to areas such as the subarachnoid space that go around the entire brain and spinal cord. So it's the fourth ventricle that is going to distribute the cerebral spinal fluid all around. Now, if we take these chambers, all these ventricles, and inject it with a plastic and fill it up, you would get a cast of those ventricles. And that is what is shown on this next slide. And I'm showing you this because we have this mold in lab for you to take a look and explore how these ventricles are set up. And so this shows the lateral ventricle, so you can see one side of it, the other lateral ventricles on the other side. You can see the intraventricular foramen, the third ventricle, the aqueduct of the midbrain, so that's leading to the midbrain or through it, and then here is the fourth ventricle. I'm going to show you a couple other views of these ventricles within the brain. All right, this is an anterior view, so we are looking head on at the brain. So this is the right ventricle and this is the left ventricle. And you can see the foramen that's going to connect the two. All right, so that foramen connects the two, which leads down to the third ventricle. There's a narrow passageway that leads down to the fourth ventricle. The fourth ventricle then leads down to the central canal, and that's going to go into the spinal cord. There is a hole, a foramen, on the top of the fourth ventricle, right here, that is going to send cerebral spinal fluid to the subarachnoid space. And I'll show you that in another diagram in the next, not the next slide, but the slide after that. I want to show you one other view of these ventricles. This is a cutaway section showing the lateral ventricles here. The third ventricle is here, and it shows you where the foramen is, and then there's the cerebral aqueduct leading to the fourth ventricle. And again, then it heads down to the central canal of the spinal cord. So I just showed you several different views of the ventricle system in the brain for you to see how the cerebral spinal fluid is flowing in all throughout the brain. All right, let's talk a little bit more about how cerebral spinal fluid is produced. So cerebral spinal fluid is a derivative of blood, and it is completely acellular. So remember when there's a word where A is in front of it, like avascular, it means without. So it is completely without cells. And it is produced in each of the ventricles. It's not made anywhere else but the four ventricles. So the two lateral ventricles, the third and the fourth ventricle. And this is how it's made. This diagram depicts that. So you will have, sorry, I already drew on that. You will have, let me change this color. You will have a capillary surrounded by ependymal cells, which are shown here in pink. And together these form the choroid plexus. And the cells are going to filter material out and transport the fluid across this barrier. And so these cells can determine the exact concentration of how much sodium, how much potassium, how much chloride, bicarb, calcium, magnesium, vitamins, or organic nutrients, oxygen, all of those things will be determined by the ependymal cells and how much of that material is getting out. So also you can transport some waste back through that through across those cells. This is rare. There's actually another place that this occurs, but you can see how these ependymal cells line these blood vessels. You can also note that these ependymal cells have microvilli. That's what's depicted here. All right, and when they are associated with ventricles, they have microvilli. Everywhere else, the ependymal cells are going to have cilia. And the cilia are there to help circulate the cerebral spinal fluid because there's no pumps to move the cerebral spinal fluid around. It is going to rely on the cilia to circulate this. So how does this get circulated? So this next slide shows how the circulation of cerebral spinal fluid is occurring. And so let's say we start to follow circulation from the lateral ventricle, which is right here. So from this arrow, from the lateral ventricle, the choroid plexus makes and circulates the cerebral spinal fluid down to the third ventricle. So we can follow it down to the third ventricle, both sides here. The third ventricle is also going to add some cerebral spinal fluid to it, so it's going to increase the volume. This then travels down the cerebral aqueduct, so we're traveling this way, to the fourth ventricle. So we're following the cerebral spinal fluid down through the fourth ventricle, which is right here. I'm trying to use a color that you can see. The fourth ventricle can also add more volume to it. Then it's going to exit out and follow through. It can go down now to the central canal through the spinal cord, and it could also enter the subarachnoid space. So once it leaves that fourth ventricle, it can then circulate throughout the brain. So it can go down the spinal cord, and then it can go back up again. And there are cilia that beat on each side. So if the fluid is moving this way, then it's going to move the opposite way. And the cilia that line this cavity are going to beat and move the fluid around. So the fluid is all going in one direction because the cilia are all beating in one direction. And this is actually a reason why we need to sleep. So it's not, the beating of the cilia is not strong enough to overcome gravity. So if you stand for long periods of time, you can accumulate cerebral spinal fluid in the lower parts of your spinal column because you're not returning it fast enough. And so lying down actually helps you circulate your cerebral spinal fluid. So even if you can't sleep, lying down allows your body to catch up. And eventually the cerebral spinal fluid will return and has to return to the venous blood supply. And that happens to be at very specific places that I'm going to show you next. So cerebral spinal fluid is flowing through the subarachnoid space, and it eventually has to return to venous circulation. And it's going to do so at the dural sinuses, which I'll show you on the next slide. At the dural sinuses through structures called arachnoid granulations. So let me show you what this looks like in a picture format first. So again, cerebral spinal fluid, as we know, is flowing through this subarachnoid space. Eventually, it's going to reach an area where the dura has been penetrated by an extension of the arachnoid membrane. And this extends into this sinus right here. And this is where cerebral spinal fluid will actually leak back out and enter into the venous blood supply. So the cerebral spinal fluid is going to enter the sinus. It's going to have access to these blood vessels right here. And once it enters the venous system, it's going to follow and flow into the jugular vein and flow back to the heart to get replenished again. So all cerebral spinal fluid will re-enter the blood through the venous system. Let me show you a close-up of how that's happening. So again, you have cerebral spinal fluid flowing in this subarachnoid space. Eventually, it's going to make its way into one of these arachnoid granulations where you can see how the fluid is then coming back out into the sinus. And there are blood vessels within the sinuses that will carry this solution back to the venous supply. So we're next going to start to look at the functional anatomy of the brain and go over those 6 regions that I spoke about at the very beginning of this lecture. And that will end up wrapping up our lectures on the nervous system.